Thank you very much for the committee and for organizing this very interesting congress. My name is Jesus Izaguirre. I've been living in Japan, as it has been already well said, and I would like to introduce my research. It's called Novel Anticancer Agent, SQAP, binds to focal adhesion kinase, and modulates its activity. This one here is the compound. I fear I'm a little bit out of my side. My research is not that related, but I will try to make it interesting. It's a derivative from a natural product which is called SQMG. It was found on sea urchins' stomachs. And they both have anti-cancer activity. This is because they have anti-angiogenic activity, and it's a radiosensitizer as well. How it works is like here and here you can see the combination of the compound plus the X-ray treatment will decrease the tumor volume. In here, there's an example with a tumor xenograft. Maybe this one is not working that well. What we do is let the tumor grow until 100 cubic millimeters, and then start the injection and the X-ray treatment. This one in here is just a compound. As you can see, the compound itself has anti-angiogenic activity, but it's not enough to arrest cancer. This is the radiotherapy, X-ray, and this one in here is the compound plus X-ray treatment. The dosage is about 2 milligrams per kilogram mouse. So this is the strategy that I have been following. My research has been on finding the target identification for SQAP, because we knew that the compound has anti-cancer activity, but we didn't know how it's actually working. I have used this assay in here. It's called phase display, and what we do, what is useful for is for finding interactions. So we have in here a library of phase, this virus here, which has up to 9 times 10 different peptides displayed on their capsids. How it works is like you pick up a target. In our case, we wanted cancer. You extract the messenger RNA from the cancer cell line, whichever you like, and then you amplify to cDNA using random primers. So you obtain very short sequences that can be injected into the virus, the FAST genome, and they will be expressed on their capsid. They are short sequences, peptides. So we have in here this library, and the other thing that you need for making this experiment is to use biotinylated compound. So you need to immobilize your compound on a matrix, on the surface of a well. So you need to have it biotinylated in order to be able to immobilize into streptavidin. So then you just add the whole library, allowed to incubate like 1 hour, and then you wash away the FAST that have not bind. The one that have remained, that are binding, you can elute them with a mild detergent and use an E. coli host for amplify them. You repeat this several times, say like 4 to 6 rounds normally is enough, and then you get the binding ones. This is the compound SQAP with the biotin in here, and we have in here a derivative glucose, propanediol, and a fatty acid chain. And these are the results. I'm trying to go very quickly, so I am just showing you the results. I got 5 candidates, peptides belonging to these two proteins first, SCP2 and MFP2. Then they were actually obtained from lung cancer and from HUVEC, which is a model for angiogenesis. These two in here, ADRN1 and HR23B, they were obtained from HeLa model, and FAK was obtained from lung cancer as well. From 16 samples, I picked up 16 random colonies and analyzed the DNA sequences of the peptides displayed on the capsid. Means like 9 out of 16 belong to SCP2. This is one of the strong points about this technique. You can actually know exactly the amino acid sequence because you can read the DNA directly, and you can know exactly the location in the protein. What's like, this is actually one of the strong points of this technique. In here, you can see that 2 different proteins, SCP2 and MFP2, share the same domain, SCP2, both of them. And another point is that, as I said, these 2 were found across 2 different phase libraries, lung cancer and HUVEC, which is shows like phase display technique could be actually quite reliable. Next thing, validation of the interactions. The first experiment that I did in here is called SPR, surface plasmos resonance. Probably you might be familiar with it. I use SQP as a negative control. It's the same molecule as SQP, but it's lacking the fatty acid here. We thought it might be interesting because the fatty acids are actually like glue. They bind to almost anything inside the body. What we're doing here is we mobilize the target protein or peptide, whichever you want, using free carboxyl groups here on the matrix. And then we flow the compound. When the compound binds with the protein, the mass change is detected by this machine and shows the response units here. These are the mass change. This is time. This is the concentration of the compound. These two in here, SCP2 and MFE2, are peptides. I produce these peptides using our E. coli expression system. You can see the concentration. As the concentration rise, the response units rise, which means that the compound is actually binding to the peptide. In this case, SQP, the negative control, didn't work, which means that SQP is binding on the fatty acid chain. These two in here, I used the full-length protein. I just bought them. Again, I could see good results as well. And finally, FAK, which is the most important target that I found. This one is a peptide. Again, I produced this one too. The dissociation constant was between the 15 micromolar up to 3, the lowest one. Then I did a docking simulation in silico assay. I used the fat domain of FAK. FAK is a very important protein related to cancer. It's actually upregulated in most of cancer cell lines. And it's also related to angiogenesis as well. So we thought it was probably the most interesting target to continue with our research. Here you can see this tyrosine 925. This is a very important phosphorylation point for FAK. And we can see that actually SQAP does bind on this point with FAK. I will explain more about this later. This is the electron map. And next, I continue these studies at cell level. This is the map of FAK. Here, as I said, this is 925 expected SQAP to be binding here. This is very important for cell motility because angiogenesis is based on the quickness of the cell about to be detached from the surface and move to another place. We thought this must be a very important target of SQAP. 397 as well is another important phosphorylation point, and we research on this, so I will be explaining about this again. We also searched some downstream targets, not only phosphorylation of FAK. For example, after phosphorylated on FAK 925, paxillin is able to bind within these residues. And the paxillin is phosphorylated by FAK. So we thought actually this SQP might inhibit this phosphorylation. And then BEGF is a final product. It's controlled by AKT, and we thought this actually must be interesting as well. Here I have some Western blot results. You can see in here how actually on a concentration-dependent manner, SQP can absolutely inhibit phosphorylation of FAK on 925 and 397. This is because after phosphorylation on 925, FAK can fold itself and block this phosphorylation on 397. Then paxillin downstream is also inhibited, the phosphorylation. BGF didn't show any important change, at least not here. This is cubic again. This is angiogenesis model we are seeing in here. More things. Right here, we were worried about the possibility that SQAP will be binding to BGF. Actually, I forgot to mention on the previous slide. For activating phosphorylation of FAK, we normally introduce BGF on the cell culture. So there's a chance that the SQAP binding BGF will block this phosphorylation. So we're using here actually SQAP with no BGF, and we can see slightly how actually the reaction was still going on. Again, then we use SQAP, which was the negative control. You can see here how does SQAP does not work on inhibition of phosphorylation, but SQAP and SQMG, the original natural product, they do both work on it. Paxillin as well is inhibited. And then importantly, AKT could be inhibited, but it needed a longer protocol. The other one was 1 hour. This took a whole day. Then this one in here is the same experiment as before, but it's done with a lung cancer cell, A549. In this case, you can actually activate phosphorylation of these cancer cells using fibronectin, but the model is pretty much the same. We just add in here and then see how SQAP can block phosphorylation of FAK. Interestingly, in this case, it was exactly the opposite. With this cancer cell model, we could block AKT phosphorylation, but not Paxilin, which took 24 hours. I'm not exactly sure why this changed, but they're just different cell lines. Again, the same experiment as before. SQAP will not block phosphorylation, but SQAP and SQAP did block. BGF did not show any particular changes in Western blot. However, we know that phosphorylation of FAK at 925 is quite important for BGF expression. Actually, there are some research suggesting that mutation on 925 will block the BGF expression. So what we did is RT-PCR in here and examine the mRNA level instead of examining the protein and the correct qubit, so a very low increase of a production of BGF. Finally, we did in here wound killing assay. It's very simple, just a scratch, a wound, and we took in here like 11 hours to see how quickly does the cells recover. We can see in here that SQAP will block this movement of the cells compared to the control. This is actually 32% against 32% remaining against 62% of the original wound remaining. Here in here it took a little bit longer, but again, SQAP makes a bigger distance than the control one. This is 46 against 68. Here's the graphics for these two, and this is like the statistics control. And then finally, we were just worried about the possibility of SQAP being harmful for the cells. So we did cell cytotoxicity check, and we could actually observe that SQAP is not, at these levels, is not killing the cells, which could be the explanation why the cells are not moving. As a summary, first display binds to these 5 proteins. Our most interesting one is FAK. And we've proved that T7 phase display is actually a good technique for studying and SPR confirming these bindings. STP2 and MFP2, both, as I said before, have a STP2 domain, are found in 2 different libraries. We confirmed this binding through a docking assay, and then the phosphorylation of FAK is reduced. And finally, interaction between SQAP with FAK is enough. Well, it's not enough, but I mean, like, it could explain why this compound has anti-angiogenic effect and is a radiosensitizer as well. And finally, I invite everyone to visit Japan because it's a wonderful, weird place, and everyone can have a good time. Thank you.
Thank you very much for your talk. Do you have any questions? Okay, I have 2 questions. One is, the first one is, this is supposed to be anti-cancer agent, your peptide, right?
Anti-cancer.
Yes. Yeah. So did you check proliferation? Check proliferation? What's the effect on cell proliferation? So
yeah. Actually, I don't have the data here, but the proliferation. Oh, yeah. As I said, I don't have the data here, but the proliferation is the SQAP by itself has not a very strong anti-cancer activity. Actually, it needs around 100 to 200 micromolar
to inhibit 50%, or?
Yeah, for the IC50. So the compound actually has to work with the X-ray treatment, radiotherapy.
Does it have to enter the cell, or you can apply it from outside?
It's actually part of it, like 60 or 70% of the compound remains on the plasma membrane, and then some of it can enter the cell.
Okay, so what would be eventual therapeutic application? How would you apply this peptide in clinics, let's say?
Yeah, I'm very sorry, but that's not my part of the research.
Or you can speculate.
I wish my professor was here now.
That's okay.
He will give you a much better.
That's okay. There is no right answer for that.
Yeah,
so
well, I could say something, but it's, as I said on the beginning, this is not my kind of thing. So I'm sorry.
That's my field. That's why I'm so aggressive. I'm sorry. Okay. Thank you very much once again. This was our last presentation for today.
Thank you.
And we'll see you tomorrow.